Okay, so today we'll be talking with Dr. Frédéric Chaux about his recent lab protocol article in PLOS ONE entitled Extraction and Selection of High Molecular Weight DNA for Long-Read Sequencing from Chlamydomonas reinhardtii. Dr. Chaux earned his PhD on plant biology in 2017 from the Aix-Marseille University in France, in collaboration with the French Alternative Energies and Atomic Energy Commission. He worked on photosynthesis in a model green microalgae, focusing on quantifying electron transfer rates in several pathways and their impact on performance and photoprotection. He then completed 2 postdoctoral fellowships between 2017 and 2022 in joint research units between the French National Center for Scientific Research and the Sorbonne University. His postdoctoral projects were on chloroplast bioenergetics and genome instability, respectively. It was during his second postdoc with Dr. Chaux that Frédéric developed the protocol we will be discussing today. In 2022, Frédéric was awarded a postdoctoral fellowship from the highly competitive Marie Curie Actions to work in the Laboratory of Marine and Atmospheric Biochemistry at the Roter Bošković Institute in Zagreb, Croatia. His current research focuses on coccolithophores, a group of phytoplankton with great impact on marine carbon cycle. Frédéric, thank you so much again for being today with us.
Thank you for the opportunity.
So just to kick off, can you briefly explain to us what the protocol that we will be discussing is about, how to use it, and why it is relevant?
So our protocol is a protocol for extracting and purifying the DNA. It is primarily dedicated to extract high molecular weight DNA, which means very long fragments of DNA. The aim is to get the longest fragment as possible and avoid as much as possible the degradation of the chromosomes into smaller fragments. So the aim of the protocol is that, and this is meant for mainly long-read sequencing of DNA.
And just to understand the significance of the protocol, when you decided to optimize and develop this protocol, what was the state of the art? Was it possible to actually obtain high molecular DNA from this species? And if so, why those protocols didn't work for you?
So for this species, actually, there was only one paper that was published, but this species was, you can probably find the references in the paper, but they did not publish how they got the DNA, how they extracted and really reach a high molecular weight, because it was one of the species that they treated in the protocol and really not their main focus. So it was actually done before, but we were not able to take the same protocol, and I had to start from scratch actually. And actually my motivation for this protocol was that I made lots of mistakes doing this protocol, and I was naively aiming at not reproducing the same mistakes over and over. And once I found something that seemed to be working well, the idea was to share it as largely as possible.
Yeah, no. So the motivation a little bit was based on poorly reporting in previous work that had already done something that was similar to what you wanted to do, but the reporting was incomplete.
Yes.
And going, a little bit deeper on the protocol itself, now that you have like a beautifully written protocol with images and very clearly detailed steps, what are the most critical steps for the user?
Yes, maybe if I may add for the significance that we spoke before, I think that the significance was also that this microalgae, Chlamydomonas reinhardtii, is a very important model for photosynthesis and for algae in general, and it has been used for decades. And the long read sequencing is also something that is starting to take more and more, be implemented more and more. So we were among the first to like meet these two topic. But it's for sure that more and more people in both communities of sequencing and in algae, in the algae and Chlamydomonas community, will need this kind of protocol.
Okay, thank you.
So, what was the question, sorry?
No worries. I was asking about what in the protocol itself, what are the most critical steps?
So for me, it was first it's a phenol chloroform protocol, so you have to be very careful with the solvents for yourself, but also it's important to during the phenol chloroform extraction to not take phenol at any moment in your sample while you're trying to separate the phases in the sample. So for what I tried to stress out in the protocol is that we start with a lot of material, and people doing this protocol should not try to get every little drop, but it's better to like waste some material and not take any phenol during the extraction than trying to get as much as possible of the sample. Because in the end, there is plenty of DNA.
I see.
So yeah.
And at the beginning of this conversation that you mentioned that you tried many things that didn't work. Can you, while you were developing and trying to get this protocol to work, can you let us know some of those things? And the reason I'm asking is because, you know, some of the users of the protocol might say like, oh, I could probably try to improve it by doing this or that, but that's actually something that you already did. It didn't work and didn't make it to the final version of the protocol.
Yes, it was a very intensive trial and many errors for this protocol. So first of it, I tried with some commercial kits, but it was really the beginning of this kind of kits for high molecular weight, and I did not succeed in having good results, especially because this alga is very different from both plants and let's say yeast or tissue, or so it does not have a kit dedicated to this species, and also it's more related to plants. The cell wall of the microalgae is completely different in composition as compared to plants, so taking a plant kit is not it does not make sense more than any other kit. So this was one part. What was important as well was that I tried the different kits and different step of purifications, but also even when you get good DNA with any of the method, it's very hard to know if it's going to be good DNA when you sequence it on the long read nanopore. And that's actually what I did. I prepared DNA from several different way, and usually it seemed quite good, but actually in the nanopore, which is very sensitive equipment, for some reason that I can't explain, the sequencing were not good, and that's why I think our paper has this added value that we also published this poor sequencing results. And especially with shorter reads. That's one of the steps that I implemented in the protocol, was to select for longer reads. And in the protocol we show how good this addition was for the high molecular weight purification.
And I mean, I know that you develop and optimize this protocol specifically to work on Chlamydomonas reinhardtii. But I wonder whether you have tried to extend it to other species, either of algae or something else. And if you have, whether you had to modify it somehow to make it work. And if you haven't, try it somewhere else with other species. What do you think would be the needed modifications for people to implement it elsewhere?
Yes, well, so yes, I used the protocol for different species. I actually used it as well for dinoflagellate and for one coccolithophore species, which are two other which are phytoplankton species. And it worked okay. I have no control with other kits or other experiments, so I can't say if it was much better or much worse than anything, but it worked well with other phytoplankton species. I think that what people should adapt in using with different species is really like the cell wall and how to get the DNA released in the media. And that was really the heart of our protocol, to be able to degrade the cell wall of Chlamydomonas. That's why there is so much of the proteinase K. And also among the different trial and errors, it was also the time of lysis of the cell, the initial step, that one could think that the longer the better, so that the cells are more and more degraded and DNA is more and more accessible. But actually what we saw was that with longer lysis time, so in our protocol it's 15 minutes, and with longer lysis time, the DNA gets contaminated by lots of different compounds that are actually in the cell as well. And it's very hard to separate these compounds and the DNA. It gets stuck for very strongly. So actually the trick for me was to have a very short lysis, starting from a lot of material, and maybe not all of the cell have released their DNA, but there will be plenty, and at least it's not going to be contaminated by a lot of other compounds.
That's a very interesting point. So, you have put a lot of effort in developing this protocol that, again, stemmed from the fact that somebody else had done the work but didn't report exactly what they did. So you had to start from the scratch. And you developed this step-by-step protocol. Now, you also got the protocol peer-reviewed. And my question is, what was the motivation to send the protocol to a journal?
Well, for me, it was really a naive opportunity to just share our working protocol. So once again, there is no very strong comparison between different protocols, lots of different things. For instance, I also tried grinding the cells in liquid nitrogen. And so my idea was to go for the simplest protocol, for the one that works. And really for other users, feel free to try and implement it and improve it. But yeah, once I got this protocol and I was able to see that it was reproducibly good enough for the long read sequencing, I think that the peer reviewed and publishing on the website was that also it makes it more available, more visible to the community.
And can you tell us how the feedback that you received from the peer reviewers helped shaping the final version of the protocol?
Well, the protocol was relatively final when we put it into the website, because I already reproducibly did it several times, several samples. This was used for another paper. Um, so I mean the ground for the protocol was really well established at the moment I decided to publish.
Okay. And I know, I mean, this is a very young paper. It was published last month. So this question might be speculative, but I wonder whether you can tell us, if any, what has been the impact of having the protocol, having the paper for your career as an early career researcher as well.
Yes, as you say, it's a bit early to to speak about it, but I think that for my career, it's like a good proof of like being able to optimize some protocols and not starting from scratch, but also uh, proposing something that works, something that is adapted to something very specific, some experiment very specific. So, and also I have got some feedbacks on the protocol itself while I was preparing the manuscript before it was published. That it was quite helpful to have this protocol. Also the presentation of the website with a really step-by-step protocol, rather than what you can see actually in material and method in an article. That's what motivated me, because I would have shared this protocol with colleagues and the community anyway. So it was good opportunity to put it on a website, so that even people that I don't know, I can't reach, can benefit from the efforts.
Thank you. Yes. And before, I don't have any more questions. So before we wrap up, is there anything else that you would like to mention that we didn't touch in the conversation?
Yeah, I might just emphasize a few things. So once again, the the big problem with this sequencing was that it was very hard for us to know before starting the sequencing whether the DNA would be good or not, even based on gel electrophoresis, based on the quality ratio that you can have on spectrophotometric measurements. Um, there are some contaminants, some things that are not detected by this method, and it was really hard to know what is a good DNA, what is not, and there was lots of sequencing experiments that went really bad with DNA that seemed to be quite good from other extraction methods. And I can't say why, and that's why it's not included in the paper, because uh, um, it's a bit hard to explain everything, and it would be highly speculative. But the idea was more like, okay, this from start to end, it works for sequencing, and it's gonna have a good yield at the sequencing step.
Well,
Yes, and there is this. Maybe I will emphasize also on the elimination of the short reads. That So in the paper, it's a commercial kit. I also tried actually to make my own solutions for excluding the low molecular weight DNA. But in the end, it was really good, really nicely reproducible with this kit. Also, it's a bit hard to use at the first time. So if users have problem with this kit, they really should try it again and again, because it's a bit delicate to use, but it works well once you really master the short read elimination.
Okay, Frédéric, I think that the technical advice that you have provided in this last section is going to be very useful for the users of the protocol. And again, I don't have anything else to ask or say. So thank you so much for your time and for your knowledge.
Thank you very much for the opportunity, and I hope it will help for this field of research.